I'm delighted to introduce Professor Stephen Neidle from University College London, where he is Chair of Chemical Biology and Research Director. Steve, together with Helen, long-term friends, has been very interested in nucleic acid structure and interactions with small molecules. He's been extraordinarily productive, internationally acclaimed. He's published over 500 papers and has contributed to, many, many different aspects of science. He's very highly recognized in terms of awards. Among his key awards, in addition to recognition in the field of cancer and structural biology, include the Aventis Prize and recently the Pfizer Lecturer in the UK. One of the research interests that we share is to try to use structural information, chemical information to design new drugs for targeting infectious diseases and cancer. And with that, it's my privilege to introduce Professor Stephen Neidle.
Thanks very much, Eddie, for that generous introduction. I really don't recognize the person who he's talking about. But we're here to recognize Helen. And I want to start off by, first of all, thanking the organizers for inviting me. It's really humbling to go back and think about what Helen has done over many years. And I'm just delighted to be here to share in the celebrations. So what I'm going to try and do today is to step back, at least overtly, from the PDB a bit and delve into almost ancient history of what Helen was doing pre-PDB, but then lead you into how all this links into much more recent activities. So this is where I'm at in London. All looks very nice, right in the heart of London. Okay. So, looking back at Helen's career, I found this as I think her very first scientific paper. And that's not bad. First author paper in science as your first paper. And what's interesting about this paper is that it's not an overtly experimental paper. It's a paper that takes data from the literature, critically analyzes it. And perhaps this was the first indication that Helen was thinking beyond individual structures. Even at the time, it was a very significant paper and has 157 citations, still cited today. But when I started off in crystallography, like Helen, a small molecule crystallographer, and in those days, in the 1960s, one had to understand the crystallography as well as the structure, as well as the chemistry. And the first paper of Helen's that I read, and the first time I really saw her name was this paper, single author paper, in Acta Crystallographica in 1970. What was remarkable about this work was that it was the determination of the crystal structure of a trisaccharide by direct methods. And in those days, determining small molecule structures by direct methods was absolutely non-routine. This was a crystallographic tour de force. If you read the paper, which I think is very instructive, you'll understand what I'm saying. And for an Acta Crystallographica paper of a small molecule to get over 100 citations, again still cited in 2013, is quite remarkable. So I was also, in my own small way in London as a graduate student, trying to grapple with direct methods. We were all doing it by hand with the aid of our very slow mainframes, and this is what Helen did in this structure. Of course, polysaccharides, trisaccharides, monosaccharides are not nucleic acids, but they're related. They're parts of nucleic acids. So the next step, the next logical step in a way, for Helen was to go to nucleic acids. And this paper in 71, when she'd already moved on to the Institute for Cancer Research in Philadelphia, was a landmark paper in our understanding of the details of nucleic acid structures. It was the first paper that described at atomic level a nucleic acid constituent of more than a single nucleoside unit. And as you might expect, Helen being an author, it more than just described the structure, it attempted to put it in the context of nucleic acids. And this was not straightforward because this particular structure exhibited a conformation that was relatively non-natural compared to natural polynucleic acids. But it was almost naturally followed by a paper looking at the conformational features of that structure and of other nucleic acid constituents as known at that time. So this was one of the very early studies of nucleic acid structural systematics, and again indicates the way that Helen thinks about structures, not just as individual entities, but what do they mean taken together? And what do they mean taken together, not just in a visual way, but in a very rigorous analytical way. So moving on to 1972, when I started to get involved with nucleic acids, I was persuaded to move to King's College London, which at that time was still a center for studying nucleic acids, not by single crystal methods, but by the well-established fiber diffraction methods. So in a sense, that was the center of the fiber diffraction universe. I wasn't persuaded that fiber diffraction had a future. It clearly had a past, a magnificent history in terms of giving us the double helix. But I felt, as others did, particularly here in the States, that using single crystal methods was going to be more fruitful, and encouraged by that UPA structure that we've just seen, many of us started to focus on using single crystal methods, constrained, of course, by the availability of material. And material from synthetic oligonucleotide chemistry was very hard to obtain. All one could really get was material at the dinucleotide level. Rather than trying to compete with what was already clearly happening in the States, I decided to look at drug DNA questions. And that was in the teeth of a very strong view in the UK, not so much here, that studying nucleic acid structure was boring. Nucleic acids were boring because all that we ever knew or needed to know was within the Watson-Crick double helix. And of course, you can call it a monotonous structure using monotony in a slightly different context. But at that time, in the early 1970s, there were real serious questions about how one could improve the then generation of cytotoxic anti-cancer drugs, the vast majority of which interact with DNA, with genomic DNA. How do those drugs interact with their targets? That was the first question. And then perhaps we could see our way ultimately to drug design in some way that we really didn't understand in those days. And all we had as a conceptual model was what you can see here, Leonard Lerman's intercalation model, which takes really a fiber diffraction type view. The model was derived from fiber diffraction, that DNA is not describable at an atomistic level, but more in terms of groups of bases, nucleotides, and a backbone which is really schematic. So moving on a couple of years, in that intervening time, my laboratory hadn't yet really succeeded in those aims of determining drug DNA crystal structures. Instead, we'd really had some luck with looking at some of the drugs themselves. And in collaboration with Alwyn Jones, who was also at King's at the time, we managed to get one of those structures into Nature. And I presented that work at a poster at the IUCr meeting in Amsterdam in 1975. And I think Helen and I corresponded before that meeting and arranged to meet. We didn't meet in that convention that you can see here, but somewhere outside the poster hall. And if you remember, Helen, that was a very hot summer. I'd come to Amsterdam with my wife and very young child. I'd left them and gone to the posters, very diligent in those days. And we got on very well. And we decided that we were interested in very similar problems. Helen generously invited me and my family to come to work in her laboratory the following year. I'm not sure that Helen quite realized what she was letting herself in for, and vice versa. But typically, generously, she said, rather than coming and staying somewhere in Philadelphia, some anonymous house, come and stay with me and my family. So we spent a summer in Helen's laboratory. I'm not going to talk about it now, another time when I've had a lot of alcohol, but it really was a great time, and it had some great scientific and other consequences. And it's started a friendship which endures still. So I said that Alwyn and I determined the crystal structure of proflavin, which turned out to be in itself very complex, which is why it got into Nature at the time. And I then went on to try and crystallize proflavin with dinucleotides. Just to show you what my Fibre diffraction colleagues along the corridor were believing were the way ahead, this type of very information-poor fibre diffraction diagrams were the best that they could get from complexes of polynucleotides with drugs. And we felt that we could do better. And we were lucky. We were lucky in getting some beautiful crystals, and that's one of them, of proflavin with our initial dinucleotide CPG. And Helen introduced me to a particular way of solving nucleic acid crystal structures, which required a lot of perspiration, a lot of insight into how the structures might be organized, and some unconventional approaches for the crystallographic methodology of the time. But it all worked, and we managed to get a very pretty crystal structure out of it, which, in a sense, I think more than fulfilled our hopes in terms of telling us about how this particular small drug and mutagen organized itself around a DNA fragment. And we were fortunate in getting that structure published in Nature in 1977. And as the title says, we thought of it, and that was reinforced by subsequent work, as a real model for how ligands interact with polynucleotides. And of course, it was followed by an analysis paper led by Helen in PNAS in the following year, and that really defined very clearly how we could take that crystal structure and relate it to how this and other drugs generally were embedded into polynucleotide way. structures. This was a very elegant paper because Helen showed in it that actually just 2 backbone angles were required for the opening up change, contrary to what was the dogma at the time. And that was followed by a number of other structures, papers, and here we've got 2 other structures which also got into Nature in subsequent years, and the collaboration really went on for many years, focused on nucleic acids and nucleic acid-drug interactions. From then, Helen further developed, of course, her interest in nucleic acid conformations, nucleic acid systematics in a much bigger arena, founded the Nucleic Acid Database, the paper in red here, and led the worldwide search and definition of a set of standards for the quantitative description of nucleic acid geometry, the paper in JMB on the bottom right. In between that, and I think she's still active in that, she's been analyzing with colleagues the ways in which nucleic acid structures, complex RNAs as well, fall into a discrete number of conformational classes. So all of that has been an enormous contribution to the literature, to our understanding of nucleic acid structure, and certainly has given the lie to those slightly uninformed protein crystallographer colleagues who condemn nucleic acid structural studies as boring. Quite the contrary. So one other small project back in 1997-96 actually sowed the seeds for a great deal of further work in many laboratories. This was the determination of a small molecule structure of a DNA binding molecule called tropsin. This binds and was presumed to bind in a very different way to the intercalators that you've seen so far, binding in the grooves of double-stranded DNA. Binding in a way that recognizes specific sequence. And the structure and the consequent complex structure determined a year or so later by Dick Dickerson provided the impetus for a great deal of activity in trying to assemble together molecules that would specifically recognize DNA sequences. And in my own laboratory, we've followed that in a particular way, trying to use crystallography to understand small molecule sequence recognition and build upon it by developing units to do that. And one such compound turns out to be a DNA recognition molecule, but also, fortuitously, having opportunistic pharmacological properties that means that it's a very interesting anti-infective agent, particularly for the hospital infection Clostridium difficile. And the molecule is now in phase 2 clinical trials. So from small seeds can grow some very interesting diversions. This year is, of course, not just Helen's 70th birthday, but it's also the 60th birthday of the double helix. And our thesis is that DNA is still capable of surprising us with its ability to do the unexpected. And I'll just give you one trivial example. I'm not sure that this is available except perhaps on the web somewhere or other. It looks like a double helix, doesn't it? But it's not. It's a fourfold helix. Now, what's a fourfold helix? Well, for the last few minutes, I'm going to talk to you a little bit about what we've been doing with unexpected DNA at the ends of human chromosomes. The ends are termed telomeres, and they comprise a number of discrete regions. First of all, there's coding DNA associated with chromatin, but that is protected by a specialized DNA sequence in humans, probably several thousand-fold repeats of the simple hexanucleotide sequence TTAGGG, together with specialized associated telomeric proteins. And so the functions of telomeres is to essentially protect the ends from unwanted degradation from recombination. The unusual feature of the DNA of telomeric ends is not only the sequence, but also uniquely that the last 100 to 200 nucleotides are actually single-stranded. They're not duplex DNA. They're not naked DNA, of course, because they're still associated with specialized single-stranded binding proteins. The way that these ends are processed in normal somatic cells compared to tumor cells and compared to every single tumor cell is totally different. In tumor cells, there's a mechanism to maintain the integrity of telomeres, whereas in normal cells, the rounds of replication are such that the ends cannot be fully maintained and the cell responds by systematically shortening whilst keeping that telomeric DNA approximately the same. Shortening until a point where it can no longer do so and the cell then shuts down replication. Tumor cells don't do that. They are capable of infinite replication by virtue of a number of factors particular to tumor cells, which I won't go into detail, but I'm really just going to focus on the DNA side. So here we've got at the top a representation of that single-stranded 100 to 200 nucleotide overhang. And it's been recognized for a long time, initially Liz Blackburn, Wally Gilbert, Aaron Klug in particular, that these sequences can form distinct non-duplex DNA type arrangements. And these arrangements we now term quadruplex DNAs. What are they? They're formed not by Watson-Crick base pairs, but by guanines self-assembling in the manner that you can see here. And the repeating unit is not a Watson-Crick AT or GC base pair, it's a guanine G quartet. And you can see a high-resolution crystal structure showing you just that arrangement. So everything assembles around G quartets in quadruplexes, and we can have an arrangement taking a 100, 200 nucleotide sequence that can fold up into, for example, the type of arrangement that you can see here, where the G quartets are the little light blue stacks of bases in the schematic on the right. And the intervening sequence holding it all together, in this instance, are the TTA sequences, the TTA loops. So we've come at a structure that's very different from the classical duplex DNA structure. And it's even more different because it has a channel in the middle of the G quartet. It's sufficiently large to accommodate a metal ion, normally potassium, can be sodium. Do these higher order structures actually exist in vivo? And this has been unclear until early this year when Shankar Balasubramanian's laboratory in Cambridge, UK, showed unequivocally using quadruplex-specific antibodies with a fluorescent tag that these arrangements do exist in vivo. And you can see on the right a magnified picture of a chromosome with the tips lighting up with the quadruplex-specific antibody. It turns out, and I'll come on to this in a minute, that these sorts of structures actually have an existence elsewhere in the genome, which is why on the left-hand side, those cell images show so many regions of fluorescent imaging. Well, we started to do crystallography in this area over 10 years ago, and this particular study was published in Nature with Gary Parkinson as the first author. Gary was an import to the UK, or re-import, I should say, from Helen's lab, very talented postdoc who was originally British, went to Pittsburgh to do his PhD, but actually did it in Helen's lab. And he was telling me 2 days ago what a great experience that was. Also being out of Pittsburgh, I think, was part of the great experience. This crystal structure is of essentially 4 repeats of the human telomeric sequence. If you cast your minds back to what I showed you a couple of slides ago, you can see that the folding is fundamentally different. It's counterintuitive. It goes up, down, up, down, up, down, up, down. It's not a smooth fold. And that produces a structure that you can see here with the TTA loops extending out from the structure and the G quartets in the middle, a propeller type of arrangement. In the middle of the structure, we see potassium ions, and you can see them on the middle view there, very neatly, a little channel of potassium ions. And that assembly happens this way. We've taken the sequence, and each quartet is formed by the donation of a guanine, 1 in each G tract. So we have 3 G quartets formed, and they all come together to form this arrangement. I said earlier, I just really went through it very quickly, that there's a fundamental difference in the way that tumor cells process and maintain telomeric ends of chromosomes compared to normal cells. And this turns out to be an avenue for potential exploitation for anti-cancer drug design. We've taken an indirect approach, trying to fold up those ends into quadruplexes, maintain them, which then turns out to be an effective way of certainly selectively killing those cells which are so affected. And several years ago, we developed an acridine compound, coming back to our old friend proflavin in a way, but giving it extra functionality. That functionality gives it selectivity to not to duplex DNA, to which it binds very weakly, but to quadruplexes. And in our laboratory several years ago, we were able to determine the co-crystal structure, which you can see here. The ligand is sandwiched in between 2 quadruplexes, as it would be in the longer natural sequence. Again, you can see the potassium ions, which are essential for maintaining the integrity of the structure. But this structure also, apart from explaining how the molecule bound to quadruplexes, provided us with further insights for rational drug design. And one of the insights that turned out to be very important, we can see how the side chains of the ligand are enveloped by the loops. You can see there on the right-hand side that the side chain surface in blue is almost completely enveloped by the surface of the 2 loops above and below. Looking at that in detail, it turned out that the loop conformations had fundamentally changed compared to the native structure, which told us, first of all, that it was dangerous using the native structure as a template for drug design. We absolutely needed complexes in order to guide us. These even small trinucleotide loops turn out to be extremely flexible and respond exquisitely to the particular geometry of a given drug molecule. So most recently, coming through to 2013, we've used the same philosophy to develop a range of further molecules. that you can see here, and the detail is in this JMed Chem paper published earlier this year. These are two different crystal structures. On the left-hand side, I want you to focus on the side chain which is embedded down in the groove of the quadruplex. And that side chain is linked to that groove through a little network of water molecules, some of which you can see connected here. In particular, water molecule that links onto a nitrogen atom on the ring at the end of that side chain. The problem with that ring is that it's positively charged and its size contributes to the bulk of the molecule. Druggability for a molecule requires the size of the molecule to be minimized, requires charge not to be excessive. So we pose the question, if we changed that from an N-methylpiperazine side chain to a morpholino one, which is much more charge neutral and slightly smaller, could we still retain the binding and the characteristics of that binding? So a second crystal structure is on the right here. And you can see that pretty well the water network is retained and the overall binding affinity, as suggested by the crystal structure, is also retained. But biologically, this is a much better molecule, has nanomolar potency against, in particular, pancreatic cancer cells, and is a subject of further studies by colleagues at University College London. What we know about such molecules, about their mode of action, is that the initial simplistic picture that we had is actually much more complex, and the formation of a quadruplex in DNA for a cell is a lethal damage signal. It leads to rapid upregulation of DNA damage and stress response genes in those affected cells, and a rapid anti-cancer effect in cells in a tumor. And that's down ultimately to the drug being positioned and stabilizing that single-stranded overhang sequence, competing off single-stranded binding proteins and forming structural arrangements of this type. I mentioned that stable quadruplex sequences exist elsewhere in the genome. Yeah, 2 more slides. And these themselves can be novel drug targets. Of course, we have to overcome the topology of duplex DNA in order to open up such a site, but we know that this can be achieved. So I just want to, before I finish, just leave you with a very recent crystal structure, as yet unpublished, of another quadruplex from the promoter sequence of a gene that's implicated in malignant melanoma, the BRAF gene. A yet more complex arrangement where now we've got 7 G quartets and a very nice continuous channel of potassiums. Again, a potential target. So I want to really just finish now by again thanking Helen, not only for the leadership role she's played in the projects that we've done together over the years, but for your profound contributions to the wider field of nucleic acid structure, and of course to the yet wider field of the protein data bank and the nucleic acid data bank. So those thanks come not just from myself, but from my family, from my wife Andrea, and from those very small children 37 years ago who now have got children of their own. Thank you very much.
In the interest of time, let's take one question.
Wayne.
What are the ligands to the ions that you said were potassium ions, and how do you know they are potassium ions?
How do we know that they're potassium ions? If it looks like a potassium ion, if it behaves like a potassium ion, it is a potassium ion. To be tried. Now, how do we actually know that they're potassium and not, say, sodium? Well, there's a whole lot of biophysics that's associated with these structures that tells us that it's likely to be potassium. But I think you're right in pointing out that one has to be careful taking oligonucleotides from, that have been synthesized for you, where there might be other ions there. So we have tried to be careful. We've sometimes done anomalous studies to try and convince ourselves that we do have the right assignments. The geometry of sodium versus potassium is actually very different because of the very tight spatial requirements. So again, we're pretty well convinced that it is potassium. Ammonium is not impossible, but I doubt it.
Please join me in thanking Professor Neidle again.